I'm really not prepared for this test. Can you help me?
Yeah, sure. What do you need help with?
So I heard that the test is focused on telomeres, and that's one of the subtopics of aging. It says that telomeres can possibly keep you from looking young. That's wrong, right?
I know it's crazy, but telomeres can really make you young.
Can you help out before we have the test, please?
Yeah, of course. So telomeres act as an anti-aging enzyme. Long story short, they're at the ends of our chromosomes, which hold our genetic info. And as cells divide, they get shorter, which means the cells eventually die, which means...
That if they don't divide and get shorter.
Yeah, so if we can somehow limit how short they get, maybe we could possibly extend our age.
Damn, that's crazy. We could be like superhumans.
Yeah, currently, I know that some scientists are working to figure this out.
Yeah, maybe. Well, thanks for your help. I needed it.
Hello, class. As you can see, we have a test today. So I hope each and every one of you prepared for the test. So take out your Scantrons and get ready to take this test. So class, does anybody have any questions before we begin the test?
What are telomeres?
Well, if you had studied, you would have known what telomeres are. But telomeres are repetitive nucleotide sequences that caps the ends of linear chromosomes. They counteract the tendency of the chromosomes otherwise to shorten each round of replication. So class, does anybody else have any questions?
Teacher Mario, I had a question. Why are telomeres important?
Teacher, I am a professor. I have a PhD, but they are important. To answer your question, they are important because telomeres prevent essential DNA from being lost to replication, and they are the basis for the number of times a cell is able to divide before the cell becomes old and can no longer divide. To answer your question. Well, class, it's time to begin your test. Okay, class, you can put your things away and I will hand out the test now. Just a word of advice, because you're going to need to know this for the test. But because DNA is only replicated in the 5' to 3' direction, the lagging strand must be replicated in segments known as Okazaki fragments. Each fragment must be primed with RNA primer through primase. Only the leading strand can be fully replicated, where the final Okazaki fragment will have its RNA primer removed, leading to the missing piece at the end of each lagging strand. Just remember that. That's why telomeres are important. You need to know this. All right, class, you may begin your test now. But I have to step outside. I need to use the restroom. I had a little bit too much fun last night. But no cheating. I don't want to see any cheating when I come back. Good luck.
This test.
Do not disrespect my test like that. Pick it up and begin your test.
So going back to what he said about what telomeres are, what is the whole ALT process?
This causes the telomeres to switch between chromosomes by homologous recombination. As a result of this swap, one daughter cell will have shorter telomeres and the other set will have telomeres.
Okay, thank you.
Why are telomeres important again? Okay, so they prevent essential DNA from being lost from replication. I think he said that because DNA is replicated from 5 to 3 end, the lagging strand has to be replicated without something called Okazaki fragments. But like each fragment has to be primed with RNA primer through primase. And then he said something about how only the leading strand can be fully replicated. Then the final Okazaki fragment will have its RNA primer removed, leading to the missing piece of the end of each lagging strand.
Okay, I'm gonna go to the bathroom and look up stuff.
Why does this question say that there's a problem with that? There's a missing piece at the end of the DNA. I'm not sure.
I think it's because there's no way of replacing the missing piece in the lagging strand. And I think with repeated replications in lagging strand, that the DNA will continually lose portions of its nucleotide sequence to the point where it can cause mutations. and cut away a portion of protein including genes. This only occurs in eukaryotic cells, which are linear, not in prokaryotic, which are circular.
Oh, I remember him saying that in lecture, this is where telomeres come in. The telomeres are added to the length of the lagging strand through the enzyme telomerase. It synthesizes nucleotides at the ends of the chromosomes by using its RNA template as a copy for the repetitive sequence. I'm pretty sure this is achieved by conventional DNA replication. The ends serve as a structural marker that defines where the ends of the chromosomes are.
All right, class, time's up. I hope you did well. It's time to hand in your test. And I hope none of you cheated, because there will be severe consequences. You hear? Good. Good morning, class. I've graded your exams. And congratulations, you all did very well. However, I know you cheated. I caught your classmate with her phone walking to the bathroom, and you all did too well for that to be a coincidence. This is all wrong. Fools! You are all fools! Like, do any of you even know how telomeres are maintained?
I'm pretty sure it's because of the telomerase.
But how?
Well, the enzyme telomerase has an RNA template with its structure that allows it to make copies of the same DNA sequence and add it to the ends of the template strand. Then I believe DNA replication takes place to make the strand complementary to that.
Well, that's correct. Congratulations. There may be hope for you yet. If you answer my next question correctly, I'll give you all 100% on your exam. So, how do telomeres contribute to aging?
You know this.
Yeah, telomerase acts as an anti-aging enzyme. When one has long telomeres, it allows to regenerate the cells to keep dividing and restoring tissue. Whereas short telomerase gives up early and the telomerase caps dwindle. Some people are lucky to have longer telomeres, but the environment has a role also. If our cells are exposed to a lot of stress and injury, even at a young age, they are focused to divide more often with shortened telomeres.
Oh, and yeah, I remember reading that at the ends of the chromosomes, there's telomeres making it possible for the cells to divide, as they prevent coding DNA from being lost during replication. When a cell divides, the telomeres get shorter, and once they are too short, they stop dividing and the cell dies. This is where the association of aging and cancer comes from.
Oh, and if you want some more information, if we were able to learn how to manipulate telomerase and telomeres, then we may be able to mass produce cells for transplantation, including insulin-producing cells to cure diabetes, for muscle cells for treating muscular dystrophy, cartilage cells for certain types of arthritis, and skin cells for healing severe burns.
Well, good job, class. It seems you deserve a perfect score on your exam. Good job.